Ecological fallout caused by the absence of starfish by Inshal Bazad, Brianna Reed, Johan Yao, Garrett Nage, and Moscon. A little background information on the sea star wasting disease. It affects starfish and several other echinoderms. The disease is associated with a rise in water temperature in some places, not all. It's caused by a denzovirus and it leads to the emergence of lesions followed by body fragmentation and then eventually death. In 2014, it was suggested that the disease is associated with a single-stranded DNA virus. Now known as the sea star-associated denzovirus, it's not fully known yet. The trophic cascades in the presence of starfish. The starfish are, like here, they're present, they are in high population. Uh, they prey on sea urchins, and then the sea urchins would prey on kelp forest, but with a lower amount, the kelp forest would increase. Um, the kelp forest here is really what we're looking at is the most important part of it. Uh, they play a pretty major role because they are a place of refuge for many organisms, are a primary producer, and also help the carbon sequestering in the environment. So, the trophic cascade in the absence of starfish, it's basically just the opposite. The sea star population decreases, that allows the sea urchin population to increase, and they are destructive grazers, so the kelp forest is destroyed.
We decided to look at the interactions between different predators of urchins, the urchins themselves, and the kelp population. Kelp forests play a significant role in the ecosystem, and its reduction or elimination altogether by the urchins can have detrimental effects to other marine organisms. The kelp forests provide a place for other marine organisms to live or just hide from predators. They're also the primary producer in the oceans, and they also help sequester atmospheric carbon into the ocean. So, the knowledge gap that we encounter regarding this type of marine trophic cascade is that there are not a lot of studies looking at the effects of the top predators that can be used to cut down the population of urchins so that the kelp population is not declining and negatively impacting the ecosystem. So, our research aims to determine whether there are other suitable species that can be used to replace the sea star in this trophic cascade in places where the sea star population is declining due to sea star wasting disease or other ecological problems. So, in order to protect kelp population from sea urchins, we propose to carry out field experiments and observational studies to look at the effects of added biocontrol organisms. In this case, we plan on using otters and see if they can help reduce the population of urchins to a manageable level so that the kelp can continue to thrive.
Our hypothesis is that locations with a higher density of otters will have increased kelp growth because higher densities of otters will be more effective at controlling the sea urchin population. To test this hypothesis, we will use a Backey experiment design. The first step in the experiment is to create barriers approximately 10 meters by 10 meters that will exclude sea stars from the test site while still allowing free movement of otters and sea urchins, as well as allow kelp to grow freely. Next, the biomass of the kelp will be measured at each of the locations. There will be between 12 and 20 locations total, with 3 to 5 at each areas of low, medium, and high otter populations, as well as a control location, which will have an otter population of 0. The final step will be to use statistical analysis to determine the effect of the different otter densities on the average biomass of the kelp.
So moving on to the study design of the experiment, removal/addition studies. Here, if we take a look at this graph, it depicts a good relationship of the species in this experiment, and we can see the effects on kelp that were observed after we removed urchins and otters. In the absent scenario here, we expect to see this kelp population thrive since there's no predators present. Adding otters and urchins in the present situation, the kelp population is expected to be down since there's urchins present, but the urchins preying on the kelp will also be lower since the otters are present in the situation as well. Removing urchins but not otters, we expect to see the kelp population significantly reduced, while high rates of herbivory by urchins on the kelp will also be depictable. These experiments are going to be carried out in areas where we are successfully able to collect data on urchins preying on kelp and what kind of effect that has in that ecosystem with the absence of a predator, in this case being the otter. Next, evaluating data. An ANOVA test will be used to test the mean of the difference in kelp biomass for each of the experimental locations. Linear scatter plots will be used to infer the general effects and correlation of absence or presence of predators on aiding kelp growth. Linear regression will also be carried out to see the effects of the addition and removal studies that we previously discussed.
In order to obtain accurate results that will measure the effectiveness of this method, it will be important to minimally alter the environment and keep track of any changes that occurred during this experiment. This includes maintaining the presence of other organisms in the area and monitoring any sort of abnormal events that may occur, like for example, maybe the decrease in water temperature or the appearance of disease. There also remains a risk that the introduced species may cause an unpredictable harm to the environment. So it would be appropriate to passively measure any major impact that the otters may have on other aspects of sea life. To finish up, I would like to quickly touch up on the significance of this experiment. First off, conducting this experiment would help us determine if it is possible to use a species to restore the ecological balance. Secondly, as we all know, human behavior as well as certain natural disasters cause a lot of disruption in the environment. If successful, this method could be a solution to help the ecosystem recover from unpredictable changes. And lastly, this method is not limited to a specific condition, and with a few changes, it could be adapted to different environments and used to fix problems elsewhere.